Hey guys, welcome and welcome back to my channel. This is Dr. Prasanjit Das, currently JR1 in the Department of Pharmacology, AIMS Kalyani, and I am back with one of my seminars. So today in this video I'm going to discuss about bioinformatics tool in pharmacology. So this is my next seminar topic, and in this presentation we will be discussing mainly about the introduction to bioinformatics, then the bioinformatics tools. So I will be discussing some of the bioinformatics tools, then the goals and field of bioinformatics, applications of bioinformatics, and finally the limitations of bioinformatics. So without wasting any time further, let's get into the introduction part. So in introduction, as you can see, basically what is bioinformatics? So bioinformatics is actually the union of biology and informatics. So by informatics I mean the use of computer and internet is the most essential part of bioinformatics, and it is used to store, then retrieve, then manipulate, and analyze the data which is related to the biological macromolecules such as DNA, RNA, and proteins. So bioinformatics mainly consists of the genome, the proteome. So these are the main things which are the main part of bioinformatics. Here you can see in this diagram. So bioinformatics is the union of biology, medicine, maths, physics, that is the statistics part, and finally the computer science, that is the use of computer and internet for doing the storage, retrieval, manipulation, and analysis of data which is related to the macromolecules, that is DNA, RNA, and proteins. And here also in the same diagram you can see the integration of biology, computer science, chemistry, mathematics in the form of statistics, physics, and the statistics, that is the statistics. All these are incorporated together so as to get the information and store the data related to the biological macromolecules. Now coming to the different bioinformatics tools which we are having. So in this we will be using several computer softwares. So the examples of those softwares are Blast, then we have Ensemble, then we have GenBank, then we have BioInform, then SwissProt, and many other softwares are there. So these are the most commonly used, and out of this the Blast software is being very commonly used. Here you can see the interface of Blast software. So Blast stands for Basic Local Alignment Search Tool. So we will be discussing about this in the next slide. Then we have the interface for Ensemble, and most of these bioinformatics tools, and here the tools which I have mentioned are always free, so this can be used for academic purposes, whereas some tools are provided only after getting the subscription. So once you get subscription to a particular tool, then you can use it further, but most of the tools are free to use. Now coming to the different bioinformatic tools. So first is Blast, that is Basic Local Alignment Search Tool. So basic local alignment search tool, in this we are having different types of uses. So basically we have nucleotide blast, and then we have the protein blast. So it is one of the simplest and easy to use bioinformatic tool, and in this we are actually doing several sorts of analysis. So the different types of analysis which we do here is basically we are comparing a particular unknown sequence of either genetic material, that is it can be DNA or it can be a protein, with the database. So basically we are comparing this unknown sequence to the known sequences database, and then this gets aligned to that known sequence which is present in the database, and from this we will be able to predict the sequence of the amino acids or the sequence of the nucleotides which is present in the unknown, that is the query DNA or the query proteins. So this is basically matching the unknown substance, that is matching the unknown macromolecule, that is either the DNA or the protein, with the already known and existing DNA and protein database. So this is about Blast. So as you can see, when you enter into the Blast, then when you either go into the nucleotide blast or you can go into the protein blast. So and when you are going to the nucleotide blast, here you can see you can enter the query sequence. So the query sequence, and then you can further compare it with the sequences which are already present in the database. So these two are the interfaces which are suggestive of BLAST, that is basic local alignment search tool. Then next is Ensemble. So Ensemble is basically a tool which comprises of the genomes of all the different vertebrates. So it is basically a database of genome of different vertebrates. Then we have GenBank. So GenBank is basically storing and archiving the DNA sequences which are obtained from large genome projects, and also the data which is obtained from individual private laboratories also. So these are being stored in GenBank. As you can see here, nucleotide. So this is also a type of bioinformatic tool. All the tools are free to use. Then we have BioInform. So BioInform is basically giving us several news and the latest updates related to bioinformatics tools. So this is that software. Then we have UniProt KB, which is nothing but the SwissProt. So SwissProt is basically a software which is used for the protein analysis. So the important protein database is present in this SwissProt. So this helps us to compare the sequence of different amino acids in the protein, in the query protein, compared to that which is present in the database. So this is also just like the BLAST. So these were some of the most commonly used bioinformatic tools. Now coming to the goals of bioinformatics. So the primary aim, that is the principal aim of bioinformatics, is to understand the living cell. So how do we understand the living cell? That is, we understand the living cell by looking into the different functions at the molecular level, and that is the gene which is present. Let it be DNA. So DNA gets converted into RNA, and this RNA gets converted into the protein. So this is known as the central dogma, and because of this protein, the physiological effects are mediated in a cell. So when we analyze all these, we are indirectly actually analyzing the different physiological activities of the cell. So hence we are understanding the living processes, that is the life processes of the cell. So basically we are analyzing the raw molecular sequence and the structural data which we obtain, that is from the DNA, from the RNA, from the protein. So this is the main principle or aim of bioinformatics. Now coming to the next topic, that is the field of bioinformatics. So bioinformatics, as I already mentioned, use computer and internet. So we are having different types of analysis which can be performed by the bioinformatic tools. They include the following. First is molecular sequence analysis, then we have molecular structure analysis, then we have molecular functional analysis. So what is molecular sequence analysis? It basically comprises of sequence alignment, that is the two different sequences, that is the unknown sequence get aligned with the known sequence which is present in the database. Then we can do sequence searching, that is in the database we can search for different sequence. Then we can use the software for finding the gene and promoter. Okay, so we can do the finding of the promoter. Then next is finding motif. Motif is basically a small pattern, short recurring pattern in a gene which is actually significantly affecting the gene. So we can find that also with molecular sequence analysis. Then we have molecular structure analysis, in which we look into the structure, that is we can look into the protein and nucleic acid structure, and then we can do the comparison. Then we can also do the classification and finally prediction of several other molecules also. Next we have molecular functional analysis, in which we look into the gene expression profiling. Then we look into the protein interaction, and we can also do the simulation of several metabolic pathways, since it is the functional analysis. So these are the three different analyses which we can perform, that is molecular sequence analysis, then we have molecular structure analysis, then we have molecular functional analysis. So these are the three main fields of bioinformatic analysis. Now coming to the applications of bioinformatics. So here you can see the several applications. So first is we have personalized medicine, that is development of individualized medicine. Then we can use bioinformatics in forensic analysis, use it in pharmacogenomics, that is the genetic variation which are actually affecting the drug responses. Then we can use it in drug designing and drug discovery. Then we can use it in phylogenetic analysis, that is finding out the relationship between the two different species who are having a common ancestor. Then we have the development in agriculture field. Then we also study the protein ligand interaction, which is also a part of drug discovery. So coming to one of the applications, that is we will be discussing all the applications one by one. So first is drug discovery. So for drug discovery, what we do is with the help of bioinformatics, we can actually find the different molecules which are binding to the receptor site of the target protein. And as a result, we get one of the target, and then the target is being validated, and finally then we can find out several molecules which can act as the lead and bind to this target protein. So this helps in lead generation very fastly, and also it decreases the time which is required for the discovery of the drug. One such example is the development of several novel drugs like imatinib mesylate, which is also known as Gleevec, which is used in the management of chronic myeloid leukemia. That is, it interferes with the abnormal protein which is formed in the disease chronic myeloid leukemia. So this was made possible with the help of bioinformatics tools. Now continuing with the drug discovery. So as you can see here, in drug discovery we are having several phases. There is first is target discovery, then we have the drug discovery, and then finally drug development, which comprises of the preclinical and the clinical trials. So in all these phases we have bioinformatics involved. So bioinformatics is used for sequencing the genome, it is used for sequencing the amino acids which is present in the protein, which is used for sequencing the different RNAs also. Then we can also use it for homology modeling. It is helpful for identifying the different 3D structure of the protein and with the help of the amino acid sequence of the protein. Then we have bioinformatics, which is also used for finding different biomarkers of the effect of the drug, then to look for the toxicity of the drug. So these are involved in the drug development process. Then we can correlate the genomics, the proteomics, and finally the transcriptomics with the clinical data which we obtain during the trials, and this will help us to ascertain that a particular drug is only responsible for this particular effect and what genetics are involved in causing several toxicity or increasing the efficacy of the particular drug. So in both the areas we need bioinformatics for managing the data, manipulating it, and finally doing the analysis. Now the next example is that in one of the early contributions from bioinformatics field to drug target discovery is the identification of a sequence homology between simian sarcoma virus gene, that is VSIS, and platelet derived growth factor. So this finding, because of this we got to know that the particular gene, that is simian sarcoma virus gene, it is having a structure which is very similar to platelet derived growth factor. So we know now the structure of this gene because we know the structure of the platelet derived growth factor, and as a result we can develop the drugs against this virus. So this was one more area where the bioinformatics has helped in the drug discovery. Now coming to the next application, that is personalized medicine. So patient genome sequencing actually detects some of the potential harmful mutations which are responsible for causing the disease in the future. So as a result, this will help in early diagnosis, and the screening can be also done. It can help in screening, plus it can also do the early diagnosis, and finally because early diagnosis is done, then we can initiate effective treatment leading to a better patient outcome. So here you can see one of the mutations which is present in the normal genome. Then next application is polymorphism research. So single nucleotide polymorphisms are valuable resource for investigating genetic basis of disease. Here you can see that in the normal population we are having this particular nucleotide, but in case of single nucleotide polymorphism, in some other individuals we get a different nucleotide which is a single nucleotide which is different, and as a result this is responsible for predisposing the individual to several diseases like diabetes mellitus, hypertension. So if we find out this earlier, we can take the preventive measures as such. So this is also one area where bioinformatics is used because it helps in sequencing the DNA, that is the nucleotide sequence. Now coming to the next field, that is it is used in forensic medicine, that is because phylogenetic analysis reports can be used as an evidence in criminal courts. Then in agriculture advancement, so how bioinformatics help in agriculture advancement, that is bioinformatics is used for sequencing the genome of the plant, and as a result when we compare that particular genome with the different phenotypes, we can help in the development of new crop varieties which are actually having more resistance to the disease, and they are also having higher productivity. So these two factors we can combine and we can produce better crops. Next is in the field of microbiology, it is also used for studying the antibacterial resistance and development of the antibacterial drugs also. One such example that is where bioinformatics was used in the field of microbiology is that bioinformatic analysis. It revealed a glutamate transport system which is present in the pathogen Clostridium perfringens, but this same transport system is absent in the mammals and the birds. So drugs developed against this transport system not only protected the humans, but it also protected the domesticated mammals and fowls. So this is one example where we can use bioinformatics for the help of development of some of the drugs which are acting against the particular pathogen but is not causing the toxic effects to the host. Then the other examples which are included here is the Giardia intestinalis. It is having a signaling pathway in it, that is the phosphoinositide 3 kinase signaling pathway. So this pathway is essential in several eukaryotes, so we need to identify the proteins involved in this pathway and what is the difference between the proteins present in the Giardia intestinalis and the mammals. So it was found that there is a special insertion in case of the Giardia intestinalis sequence genome. So that was being targeted as the area for the treatment, and as a result this also helped in the development of the drugs which are effective only against the Giardia intestinalis and did not cause any toxic effect to the humans. Similarly, we also had drugs which were developed for the management of infection by Pseudomonas aeruginosa. Now coming to the limitations of bioinformatics. So we can say bioinformatics is actually a field which is having a great number of inherent limitations. So these limitations include the following. First is we need actually high quality data in the process of in the process of this drug discovery, or be it development of personalized medicine. So if the data quality is poor, then it will lead to improper analysis. Next is bioinformatics cannot replace the traditional experimental research methods, the normal experimental research methods, that is the trial and error method for testing the hypothesis. So this is one more disadvantage of bioinformatics. Next is the sequence data which we obtain by high throughput screening, that is high throughput analysis, which we are sequencing further. If it is error prone and if it is having more number of errors, then it will lead to the development of errors in the downstream analysis also, which is done by bioinformatics, as it is going to process the data which is already containing the error following the high throughput analysis. So these are some of the limitations, that is bioinformatics require high quality data. Next is bioinformatics cannot replace the traditional experimental research methods, and then the sequence data which is obtained from the high throughput analysis can obviously contain several errors which will lead to further development of errors in the downstream analysis. Now coming to the conclusion. So bioinformatics basically apply in silico analysis and solve biological problems using mathematical and statistical techniques, and the bioinformatics tool also detect the associations between the genotypic and the phenotypic data, and it helps in advancing the translational sciences, that is basically applying the knowledge which we obtain from the genomic studies in health and improving the health care of the patients, and thus finally also help in the development of personalized medicine. So this is the final conclusion of bioinformatics, and these are the references which I have gone through for making the presentation. So I hope you like this video. So if you like my content, please do like, share, subscribe, comment, and also press the bell icon to get notified whenever I upload a new video. Thank you.